So now we are broadcasting on Zoom and Facebook. Yeah, I think that there was a Seattle chapter. I'm not sure how active they are at the moment. If you are interested in it, then I'll put you in contact with our fearless leader in Florida and help you get started. Or if you know people,
you can start your own chapter actually.
Yeah.
Really? That's pretty interesting. I certainly would like to learn more about that. Just taste of science in general, you know, I don't really know too much about it.
No.
Yeah. Well, I hope you bought a drink with it.
Right,
yeah, like a kratom and coffee tea, basically.
Or, as
That
our
sounds
subtitles say,
interesting.
a crayon and coffee tea, which has more of a waxy composition.
Yeah. Mm.
I'm sure it makes you wax poetic. First pun of the event.
You can taste the colors. It's a good synesthesia.
Shiny, colorful tea.
Mm-hmm. Bringing new meaning to taste the rainbow.
Yeah.
Okay, guys, we're about to go. I'm going to disappear since I am not a panelist or moderator, but have fun. I'll be behind the scene if you need me.
Awesome, thank you.
All right, we have 19 attendees on Zoom. For the people that are just joining us now, we'll be starting around 7 o'clock. Right now we're just having friendly conversation. If you have any questions you'd like to ask us, or go ahead and get them into the Q&A or toss them into Facebook chat.
Is there a bit of a drought in Texas too right now?
Not at all. Well, not in Southeast Texas.
Lucky. I don't know if you guys have heard, but the weather's not looking good here right now.
I've heard, yeah, the burn season is expected to be pretty bad.
The burn season already started about
my mistake.
Early. Fires have already been started up. It's like we've been setting records on heat and rainfall, like lack of rainfall already. So we're looking at a pretty historic drought. And you know, we all thought fire season last year was bad, but we'll see.
Yeah, I recall the video, the Blade Runner looking video from some of the quadcopters flying around the cities. That was, you know what, we get flooding here. I think I'll take water over fire.
Yeah, I would much rather have like flooding than fires. I mean, it's pretty disorienting. I don't know if you saw photos of last year, but you know, the like skies were red. Like there were days when I woke up in peak fire season and it was surreal. Like just not even like kind of a faint sunset, pink red, but like red, like hellish. It was pretty spooky stuff. And it's looking like we're lined up for a similar, if not worse, year. So
it is most decidedly not lit.
I know everybody was joking about like with the burns last year, how good morel season would be this year, but we're drying out so quick now that morel season's getting cut short even. Just too dry.
So the moral of the story is to water your mushrooms?
Oh, Lord.
Say again?
So the morale of the story is to water your mushrooms?
As much as possible, yeah.
To our audience, if you don't like puns, you should just basically leave right now.
Yeah. Hey,
So,
are you guys familiar with the concept of Witzelsucht?
Yes.
So
I'm afflicted.
I think a few of us are probably, you know, suffering from mild cases of it. I actually spent a whole morning one day when I was supposed to be working on a book chapter with my co-author. We just spent the whole morning making puns. It was horrifying and gratifying, and yeah.
Incredible. They call me the Punisher.
The pun what?
The Punisher.
Pun.
Oh man.
All righty.
49 people. Should I go ahead and play us in?
I think we're
Yeah,
good.
we probably have some Facebook folks too, right?
Oh yeah, definitely.
Your fight or flight response.
Taste of science.
Tectonic weapons.
Nuclear weapons.
Taste of science.
Earthquake gun, I guess? Why is it on the internet?
Taste of science.
With flesh-eating bacteria, brain-eating amoeba, and things like that.
Taste of science.
They have to be saved by whales.
Taste of science.
No neuroscientists on our end were harmed with the making of this movie, at least not permanently.
Taste of science.
We are 90% of our brain away from becoming the universe.
Hello and welcome to Taste of Science's annual festival, fully online this year from now until May 1st, when we'll have a fun trivia night based on all of our events. So Taste of Science is a volunteer nonprofit organization with local chapters dedicated to fun and accessible science outreach for fellow sci-curious people. Our Houston chapter also has a new podcast, of which the theme song you just heard, called Tosh After Dark, where we discuss the science or lack thereof in various films. You can find us at tasteofscience.org or on social media at tasteofsci and at tasteofsci.hou. And our chapter's podcast is at anchor.fm/toshafterdark on Spotify and Google Podcasts. Grab a drink if you haven't already, because zoinks! Today's event is Breaking Bud, the science of drugs. Like, how do drugs work, man? Our knowledgeable speakers will answer this question and more. Stay tuned for our Q&A at the end of the event, where you could win a prize for asking a good question. Our speakers will answer your comments and questions from Zoom and Facebook Live. And now introducing our first speaker, Dr. Spencer Green is a medical toxicologist who will be discussing the pharmacology of psychedelic mushrooms, Jimson weed, morning glory, and mescaline. He will also be examining these plants' clinical features and how they're used in medical treatments. Hello, Spencer.
Hi, happy to be here. So I'm actually not going to talk about how they're used in medical treatments, but rather how we treat people who are under the influence. So I'm happy to be here. It's nice to talk about something other than snake bites for a change. But I have to confess, although I've treated many of these conditions, I've lectured nationally on this topic, I've published on this topic, I have literally no subjective experience with any hallucinogens. For better or for worse, I can honestly say I've done no hallucinogen, no recreational drug in my life. Thank God for alcohol and kolaches. So I do have some vices, but I have no firsthand knowledge of any of these. So I'm going to share my screen, and like Ariel said, I'm going to talk about 4 cases. These are all real cases or amalgams of cases, and talk about, so can you guys all see that? Excellent. All right. I'm going to talk about, what these people look like under various influences and what we do about it. So this is going to be super casual, and there's only 30 something slides, so it shouldn't take too long. I'm happy to answer questions, when it's convenient. So getting high on Mother Nature's dime, a 4 for 420. So again, 4 cases of natural hallucinogens. I'm going to start with one of my favorites, one I've actually seen so many times. So the case I'm going to present is just one of many similar ones. So I'll let you all read. You can see it's a 16-year-old who was just out of his mind talking about ninja coyotes. Actually, I changed it to coyotes to fit the whole Southeast Texas thing. The real case was ninja javelinas, because this was in Tucson, Arizona. And he was fine until he wasn't fine after drinking this tea. And this is a true story. In the emergency department, he started to masturbate, and he was really uncoordinated, so I kind of felt bad for him, because I know what he was trying to accomplish, but he didn't have the dexterity to do it. So as you can see, his vital signs, for those who are unfamiliar reading a medical chart, his blood pressure is 150/94, which is a little high for a young, healthy guy. And he's tachycardic with a rate of 121. He's breathing fine. His temperature's okay initially, and his oxygen saturation is great. And on exam, he has really big pupils that are not reactive. And that's a clue to those of us in medicine that it's a particular toxidrome. He also is very dry and he has no bowel sounds and his skin is hot and red. But the thing that I like best as a toxicologist, he had a unique speech pattern, a mumbling speech, which we don't see in any other real condition. We only see it in the antimuscarinic toxidrome. And you may have slow speech, but the mumbling speech is unique. And in this case, he had an antimuscarinic toxidrome from consuming Jimson weed. So Jimson weed is one of the more common names for multiple plants in the Datura genus. And you can see it's also known as devil's trumpet, devil's weed, thornapple, stinkweed. And until I moved to Texas, I had this everywhere I worked. In fact, sometimes it was in close proximity. At one of our poison centers, it actually grew outside the poison center. So we used to see this a lot. If you've never seen it clinically, you may have seen it on television because I think it was season 4 of the TV show ER, where a kid came in after drinking a tea with similar signs and symptoms. So the way this works, if you think about your autonomic nervous system, you have your fight or flight drive from your sympathetic nervous system, where you're ready to go, ready to fight, ready to get out of there. But then you have your feed and breed system, your parasympathetic, that allows you to do reproduction and the basic things that you need to do when you're not running for your life or trying to kill someone. And these are mediated by acetylcholine, and the end organ targets are the muscarinic receptors on these organs. So when you stimulate parasympathetic nervous system, you get salivation, you get a slow heart rate, bradycardia, you get a little bit of bronchoconstriction, you stimulate your guts, you allow yourself to urinate. The toxins found in Jimson weed are various tropane alkaloids, so atropine, hyoscyamine, scopolamine, and others, and they're found in different ratios, and at different times of the plant life cycle, you have more or less. They're found throughout the plant, but primarily in the seeds. A seed may contain as little as 0.05 milligrams of atropine. It may contain up to 0.1 and occasionally 0.2. And these tropane alkaloids antagonize these muscarinic receptors. So we get an anti-muscarinic toxidrome. You'll also hear people call it anticholinergic, but I find that kind of vulgar and non-specific, so I try not to use that in any scholarly venue. Actually, I try not to use it at all, and I chastise my residents when they use it because it's non-specific. So we antagonize these things, so you're not getting the salivation. Instead, you get dry mouth. You're not getting active bowel sounds. You get an ileus. You're unable to urinate, so you get urinary retention. And you get these big pupils that can't respond, as opposed to like when you're on, say, cocaine and you have big pupils that do react. These don't react. So the clinical features, they have delirium. Now, sometimes they're kind of out of it. Sometimes they're just crazy out of their minds. They have that unique mumbling speech. They have the mydriasis, the big pupils that don't react. Now, interesting, there are some medications that are antimuscarinic, but have tiny pupils because they affect other receptors, primarily peripheral alpha receptors. You get tachycardia. Blood pressure is usually a little elevated, but sometimes, either if they're volume depleted or the heart rate's so fast, they can actually get hypotensive because they can't generate sufficient stroke volume. They'll be hot, they'll be red and dry. Like the classic mnemonic is hot as a hair, red as a beet, dry as a bone, mad as a hatter, blind as a bat. Again, no bowel sounds and they'll have urinary retention. Frequently we see seizures, and that's probably multifactorial, and often they sustain trauma when they're under the influence, and that's how they come to our attention in the room departments. They are living for naked city, probably because they're so hot and because of the delirium. So oftentimes they are in various stages of undress, which again is not unique to Jimson weed or antimuscarinic toxicity. We see it with PCP, we see it with other things. We see it with some of my colleagues here at Taste of Science on the weekends, but they do come in naked frequently. And typically they use anywhere from 10 to 100 seeds, which again have 0.05 to 0.1 mg of atropine or other tropane alkaloids. This is a subjective experience from the Erowid website. I'm sure many people are familiar with this website, but it is a huge repository of all things related to natural hallucinogens. I've actually had the opportunity to meet Fred and Earth Erowid when they spoke at a conference I hosted in Phoenix back in 2014. And it's just a wonderful website. It's erowid.org. I think if you type in erowid.com, it'll reroute you there. And this is just one of the subjective experiences. And I'll let you read for yourselves. But people have this unique experience after using it at the appropriate dose. Obviously, if you use too much, you're too delirious to explain. So what do we do about these people? You want to keep them safe. Again, they often incur injury while under the influence. So we want to keep them safe. We want to keep them cool because one of the potential life threats is that they get hyperthermic. So we have to fluid resuscitate. We have to actively cool them sometimes. And of course, they can injure themselves and others when they're agitated. So we need to chill them out. And for Jimson weed and other antimuscarinic toxicity, we actually have a specific antidote called physostigmine. I'm not sure who is and who is not on this presentation, so I don't want to go into how much I love physostigmine, but feel free to contact me later. But suffice to say, I love this antidote very much. It's very effective, and unfortunately, we're running out of it. So in the near future, we may have to approach these intoxications with a different way, but we want to chill them out. And the way we chill out other people, not as effective for this particular toxidrome. So although I love benzos personally and professionally, they don't work especially well for these patients. All right, I've got another case. So this was someone also from Arizona, and she was using blue saucer brownies. And as you can see, she's a little tachycardic, but not particularly. And she's just watching her hand move back and forth, and her pupils are big, but they are reactive. So blue saucer, what is this? So this is Ipomoea violacea. This is Morning Glory, not named for the horrible cartoon, the horribly offensive cartoon that has been around for years. This is one of about 1,000 plants in the Convolvulaceae family, whatever. There are similar ones, such as the Hawaiian baby wood rose, that cause a constellation of hallucinogenic effects that I'll describe in a second. Some of the more common names for this morning glory include heavenly blue, blue star, pearly gates, and flying saucers, partially because of how they look, partially because how they make you feel. And they contain two types of toxins. They have LSD analogs. So they have lysergic acid amide and lysergic acid hydroxyethylamide. And those are primarily serotonergic. In fact, many of our hallucinogens, as well as many of our pharmaceuticals designed to improve mood, mediate their effects through serotonin. I like to think of serotonin as the consultant of the neurotransmitters. Serotonin has no primary responsibility, but it sort of tells everybody else how to do its job. And when it comes to mood, it really does affect a lot of things. And LSD was essentially a uniquely serotonergic agonist, and Morning Glory does primarily that. But it also has some ergot alkaloids. It has ergonovine. And you may remember ergotism is something that can have both central effects and peripheral effects. And the peripheral effects are really nasty in that they can cause vasoconstriction so severe that it can cut off blood supply to your extremities. So what happens when you use this? You get a variety of distortions. They can be visual, they can be auditory, they can be olfactory. And sometimes there's synesthesias, sort of a confusion of the senses. And I like to describe, actually, I think the best way to describe synesthesias is a song by, what's her name, Nina Gordon, who had been the lead singer of Veruca Salt. She sang in a song, I want to build a camera custom made to tape record the smell. So when you want to take a picture of something capturing the sound of a smell, that's synesthesias. So these people, they're not usually particularly agitated. They have these distortions, they have these hallucinations. They're often aware that they're having hallucinations. Sometimes they'll have some GI effects. In large use, they can have serotonin syndrome, which can be characterized by agitation and confusion, as well as some peripheral manifestations such as tachycardia, high blood pressure, diaphoresis, which is profound sweating. The other thing that's kind of neat about Morning Glory is they have really bad flatulence. This has been documented in literature. This is not just my experience taking care of farting hallucinogen users. It's documented in the literature. They have a lot of flatulence. I'm not sure why that is. Typically, they use 25 to 50 seeds. Well, I shouldn't say typically, frequently they use it, but some people do require more, anywhere from 100 to 400 seeds that have the various concentrations of the LSD analogs and the ergonovine. And again, we have some subjective experiences from Erowid. Here's one more. Enhanced colors and edges, no movement or trails to speak of. So without losing your noodle at all, that's important. You don't want to lose your noodle. That's what my urologist always said. Okay, how do we treat these people? Again, supportive care. Keep them cool, keep them calm, keep them safe. Benzodiazepines are God's gift to emergency physicians and EMS providers, and benzos work really well when they're agitated. Otherwise, you can just put them in a nice, cool, quiet place. And incidentally, I realize anyone over the age of 30 will appreciate this photo and know what it comes from. Anyone under 30 will be like, what on earth is this? Why is it appearing throughout this lecture? Well, that's your homework. All right, we're halfway done. Case 3, spirits in the material world.
I'm so sorry, Spencer, just real fast. Can you stay a little bit closer to your mic, please?
Yeah, sure.
Thank you so much.
All right, can y'all hear me? Give me a thumbs up. Okay, so this was a kid I took care of in Phoenix. I felt really, no, wrong kid. This is a kid I took care of in Texas. So this guy was wandering through the desert. No, it must have been Phoenix, I guess, looking for a religious experience. This may also be a kid who wandered through the set of Young Guns, because this happened in the movie as well. So he was confused, a little agitated, and he was covered in his own emesis. And you can see he's hypertensive, especially for a 15-year-old. He's a little tachycardic, he's hyperthermic with normal respirations, normal oxygen saturation. He's got big pupils that are also reactive, and he's very sweaty. He was using peyote, from the cactus Lophophora williamsii. And this is something that's used in, churches of Native Americans or Native American churches for religious ceremonies, and used for non-religious purposes by people who have access to them. And they come in buttons. Each button has about 45 milligrams of mescaline, and they typically use anywhere from 270 to 540 milligrams. So what's that? 6 to 12 buttons. And mescaline has the structure you can see on top. Now, it's funny, if you told me 15 years ago that a lot of my career would be devoted to structures and recognizing them and what they would do, I would have thought you were crazy. But when I look at this, I see this, phenylethylamine. So you have a benzene ring, and then you have 2 carbons, and then amine. That basic skeleton tells me it's a stimulant. And then, as you can see, we have 3 what we call methoxy groups to the left of the structure. And the more structures you have on this benzene ring from the phenylethylamine, the more serotonergic effects you get. So a basic phenylethylamine, you'll get stimulant effect. In fact, amphetamine is just essentially the basic structure plus an extra methyl group. The more substitutions you get, the more additions to the benzene ring, the more serotonergic effects. So in addition to the stimulation and the agitation, you'll get the hallucinogens, the synesthesias, etc. And you can see, oh, actually, there's the amphetamine structure right down there. Didn't even realize I had it. That was smart of me, huh? How about that? So that's mescaline. It has a combination of stimulatory and serotonergic effects. So clinically, depending on the ratios and how susceptible a person is, he or she may have a lot of agitation. They may have tachycardia, hypertension, mydriasis. They may be agitated. They may have seizures. And they often have vomiting, because to get the effect, you typically have to consume enough of the mescaline that's going to induce vomiting. And again, in the scene from Young Guns, they all vomited before they found their spirit animals. Here's a subjective experience. I find these very entertaining and enlightening. And how do we treat them? Again, supportive care. Keep them cool. Chill them out literally and figuratively. And keep them safe. Don't let them hurt anyone else. Don't let them hurt themselves. All right. The last case I'm going to do today, before I turn this over to one of my colleagues, the fun guy. And this was a guy I took care of in Phoenix. So this guy was using psychedelic mushrooms, and he probably wouldn't have come to our attention but for the fact that he fell off a cliff while chasing his shadows. So he came in as a trauma, but his whole clinical feature did not look like primarily trauma. It looked like there was something else going on, and it's because of the psychedelic mushrooms. And as you can see, he reported seeing all the warm smells. So again, synesthesias. And there are a variety of psychedelic mushrooms that my colleague Sydney's going to talk about in a few minutes. And what makes these psychedelic, they have psilocybin. And you can see there are a number of species from multiple genera that have psilocybin. And psilocybin, as you can see here, gets dephosphorylated to psilocin, which is a serotonin analog. And again, serotonin, sort of our neurotransmitter consultants, that has effects on moods and your sensations and your interpretation of external stimuli. So these people, they typically use 5 grams, and you can use it in a variety of ways, and I'll let Cindy talk about that. And when they use them anywhere from 15 to 60 minutes, they'll start to have euphoria, a variety of illusions and hallucinations. They're typically aware of what's going on. They may be hyperkinetic, so they may have a lot of psychomotor agitation. They may have tremor, ataxia. Sometimes they're tachycardic, often they're not. They may have bronchitis, and they may have some anxiety, and less commonly agitation. So here's a little Moby-themed subjective experience. And again, finally, treatment is supportive. There's no specific antidote. Put them somewhere quiet and safe. The symptoms usually peak in the first few hours, and then they get better over the next few hours. And some have some lingering effects for a while, which again, I'll let Cindy talk about. I just want to leave you with just some random mushroom thoughts. At this time, I'm going to either answer questions or turn it over to Cindy, depending on what Ariel wants me to do. But thank you for listening. Thank you for being here tonight. And enjoy 420 responsibly.
Thank you so much, Spencer. That was so fascinating. We'll save our Q&A for the end. Make sure to put your questions in the comments on Zoom or Facebook Live, depending on where you're at. And our next speaker is mycologist Sydney Ober-Singleton, who will talk about psychoactive fungi, how they grow and how they evolved. Additionally, Sydney will discuss the 2 major groups of psychedelic mushroom containing psilocybin and isohoxol, as well as their range and habitat, the variety of compounds they generate, and the biological and ecological function of those compounds. Sydney?
All right, can y'all hear me? All right, thanks for having me. I'm gonna go ahead and share my screen, get a slideshow up. All right, so there's a remarkable variety of psychoactive mushrooms out there. And part of that is just due to the insane chemical diversity within fungi. But you know, psychoactive compounds aren't limited to a single taxonomic group. They're not limited to 1 or 2 species. There's a number of different compounds, from like psilocybin, psilocin, to even DMT produced by some fungi. You have like isoxazoles, the whole class of ergotamine alkaloids produced by ergot, and a few other fungi in Clavicipitaceae. And then somewhat recently, the discovery of a salvinorin-like compound in Rhodocolibia. There's just an enormous diversity of fungi, and the chemicals involved really just follow suit. Today, I'll just be talking about really the 2 major groups of psychoactive fungi, the ones with kind of the longest history of cultural use, current use. And really that'll be the psilocybin-containing mushrooms, as well as isoxazole-containing amanita. We'll get started with psilocybin. So when you're thinking of magic mushrooms, what most people buy, consume, what the recent decriminalization and therapeutic legalization efforts have been aimed towards, is the application of psilocybin-containing mushrooms. Psilocybin is the dominant alkaloid within this group. I'll go into the chemistry a little bit more in a second. But it's found in an enormous variety of mushrooms. There's about, depending on where you stand on the taxonomy, 11 or 12 different genera that these compounds are produced by. The role of these is still under investigation, but the vast majority of these can be used as psychoactive mushrooms. They're distributed basically across the entire globe. There are very few regions that don't have some psilocybin-containing species. You know, the dominant one is by far Psilocybe cubensis. And that is like the iconic classic psilocybin containing, blue staining, dung loving mushroom. But that's really just a tropical, subtropical species, warmer climate. When you get up north, there's a number of wood loving species that grow on wood chips, also in the genus psilocybe or psilocybe. And then there's Gymnopilus is a genus with a number of psilocybin containing mushrooms. Luteifolius, and the Luteifolius group is really a big one. That's just beautiful purple mushroom, stains green when you handle it, also due to the presence of psilocybin. And then there's just a smattering. Haneolus cinctulus over here occurs in Texas and much of the US. Another dung-loving species, but you'll find it on wood chips too. Inoside, Pluteus. This lichen in the first shot contains psilocybin as well. So really, it's all over the place. This is a range map of Psilocybe cubensis, kind of showing that tropical, subtropical, warmer climate distribution. Despite what people might try to tell you, you can't find it virtually anywhere else in the US outside of people's closets. But the major two alkaloids involved, and I think Spencer might have grabbed like the exact same images for his presentation. But the major two compounds involved are psilocybin and psilocin. And these are both, well, psilocin, like Spencer mentioned, is a serotonin analog. I have this image up just so you can see really the distinct structural similarities, and much of its activity stems from the ability to interface and dock with the 5-HT2A receptor that serotonin interacts with. And there's a number of different activities involved. It can get involved in reuptake. There's a number of effects on a neurotransmitter level, and that's responsible for the huge range of symptoms or experiences you can get out of it. Psilocybin is a dominant alkaloid that you have just in a fresh mushroom. Psilocin is present in smaller amounts, but as soon as you consume it, psilocybin gets dephosphorylated. It loses this group pretty rapidly after ingestion, and psilocin is responsible for the enormous majority of the effect. And really it's a dose-dependent effect. At smaller doses, you can get, slight euphoria, mood elevation, kind of increased sociability, maybe a tiny bit of sedation depending on dose. And there may even be some serotonergic effects going on at sub-psychoactive, sub-noticeable effects that can even have long-term impacts. That's where a lot of the interest in microdosing comes from, which is largely those sub-psychoactive doses. But the content in dried psilocybe cubensis, and this is very variable, but is roughly 1%. Your sub-psychoactive dose is usually somewhere below 0.5 grams, below 500 milligrams. There's a lot of variables involved. But you get into the more like classic magic mushroom experiences, usually at like 2 grams and above, depending on the species, with something like 5 grams being a very intense experience. But at higher doses, you can get a complete disconnect from reality. And there's kind of a spectrum in between with visual hallucinations, tactile sensations, changes in a lot of what you're experiencing. And synesthesia is one thing that can be observed at higher doses, but really it's comparable to a lot of the other serotonergic classical psychedelics. You got LSD is probably the closest pharmacologically, but mescaline, a number of the others as well. And so I think that about covers it for psilocybin and psilocin. There are some other minor alkaloids, aruginacin, baeocystin, but their activity is less understood, less looked into. That's going to be an area of developing research. And then the other group I wanted to talk about are the isoxazole-containing amanita. This is a mushroom that everybody has seen, even if they don't know what it is. Your classic red mushroom with white spots, as iconic as the marijuana mushroom. This thing has a long history as an icon, as well as potential historical and traditional use among like Northern European and potentially North American societies as well. Also an area of active research, but this species occurs. It's a cosmopolitan species. You can find it in Texas and basically the rest of the US. There's some regional variation, so it comes in a wide range of colors. But the entire taxonomical section, Amanita, so Amanita section Amanita, the majority of mushrooms in that group all contain the same 2 isoxazole compounds. But really, this one's easy to identify. It's widespread. And so when I'm talking about use of the group, this is really the one to go to. It's the most well-researched. The raw mushroom contains 2 compounds that are responsible for its psychoactive effect. You have ibotenic acid and mucimol, not to be confused with muscarine. That's an incredibly common confusion, even among healthcare providers. It's unfortunately quite widespread, and it also doesn't help that it does contain trace potentially subclinically relevant quantities of muscarine. So mucimol and muscarine are both there, but it's important to keep in mind that mucimol is the vast majority of effects you're going to see from consuming it, either in a deliberate or poisoning context. Ibotenic acid, similar to psilocybin, converting to psilocin after consumption, ibotenic acid goes through a decarboxylation either before, if you prepare it, or after consumption, where it serves as a prodrug to mucimol. There's a lot of variations on preparation methods, but because ibotenic acid is known for less desired psychoactive effects, ibotenic acid's implicated, while it is psychoactive, it's implicated for causing more nausea, causing headaches. There's some evidence for neurotoxicity, yet to be studied well in humans. And generally, most people will go through some method of preparation to decarboxylate into mucimol prior to consumption. And that's really, there's a belief and potentially evidence that that significantly lessens the negative effects you get after eating it. And that decarboxylation can be as simple as heat. There's evidence it might just happen over time or even enzymatically during drying. But this is something that, again, needs some study to fully understand. Now, mucimol is a GABAergic compound. It is not really comparable to many other drugs. Some aspects of its activity are poorly understood, or at least not outrageously well studied and conveyed, largely because at higher doses, it can cause hallucinations, changes in like tactile perception. An interesting side effect that some people experience is auditory hallucinations, changes in the perception of size of objects. And similar to psilocybin and psilocin, it also has some dose-dependent effects. You will see kind of a drowsy, almost alcohol-like experience at lower doses, maybe some sedation, and some people get altered dreams, actually. Interaction with dreams during sleep is a very common report. But at lower doses, people will use it for things like insomnia. Higher doses is when you get pretty severe intoxication, somewhat comparable to really, really intense like ethanol intoxication, like really, really drunk, is how a lot of people describe it. You'll often get drowsiness that you just cannot fight. Both at high doses and lower doses, people often report that they have to sleep or they just fall unconscious. Often a very interesting dream-filled sleep. And then following that, you'll wake up potentially with euphoria, hallucinations, again, dose dependent. But really it's important to distinguish that muscimol is a lot different from psilocybin. That's a pretty common confusion, as just people think like magic mushrooms and they think that there's a comparison to be drawn. Completely different monsters, chemically and in terms of the experience. And muscazone is just another interesting metabolite and degradation product that you'll get with UV. Don't know too much about the activity there. But then the final thing I wanted to cover before I hand off is the potential role of some of these compounds. It's overall anti-predatory, like anti-predation, anti-herbivory functions of many different compounds, both mycotoxins and just other metabolites in mushrooms in general, is very poorly studied. Evidence-based conclusions on how some of these compounds might repel attackers are overall just absent or poorly founded in general. You know, it's certainly a strong theory that these things play an anti-predatory role. It's one of the only functions we can foresee for a number of these. But again, it's something that we really need research for. And the profound activity and potency of some of these compounds, not only that, but the widespread production of things like psilocybin, for example, really does hint at a crucial role. Psilocybin is a great example of this. This is from a great paper on suspected horizontal gene transfer of the gene cluster for producing psilocybin and psilocin. Like I mentioned before, the compound's produced by something like 11 or 12 different genera, and it's not really congruent. You know, there's pretty wide phylogenetic distance, yet the same gene cluster is seen all over the place. Horizontal gene transfer is a very rare event in most contexts, and to see it transfer repeatedly to so many different groups and then also get maintained up until now really does support that it has some crucial role. And with psilocybin, for example, it's kind of funny, you know, the suspected role is anti-predatory, potentially anti-invertebrate, but it may not be because of the psychoactivity the compound possesses. A recent paper came out on the bluing phenomena when psilocybe are damaged, and found that there's the enzyme catalyzed formation of these polymers, more or less. You'll get polymerization of psilocin into these giant 13-mers, and they found that when simulating the conditions of an insect or an arthropod GI tract, that they pump out damaging reactive oxygen species. So despite the profound psychoactive properties of psilocybin and psilocin, it may actually not be related to the final function of the compound in an insect gut. It may serve to blow holes in their GI tract with reactive oxygen species. But again, needs investigation. Isoxazoles, so ibotenic acid and muscimol are also potently insecticidal, just as is. So there's pretty strong support there. The common name fly agaric actually comes from its traditional use as an insecticide. You know, as the story goes, you chop it up, drop it in a bowl of milk, and the flies will drink it and die. And I've actually seen this executed remarkably effectively with Amanita pantherina in South America. It was fly season there. A little anecdote, fly season there. He set out his bowl and then later posted videos of hundreds of dying and dead flies within about 5 feet of the bowl. So these flies were feeding, eating, like drinking the milk, and then dying before they could even leave the room. There was intoxicated flies everywhere. Remarkable. The only time I've seen it implemented to that effect, but certainly the name fly agaric is not unfounded. But the bottom line is we really do need to investigate the role of a lot of these compounds, why they evolved, and why they would be so effectively transferred to many different groups. I think that about sums it up. I'm happy to take questions when we move on to that section, but I'll pass it on for now.
Awesome. Thank you so much, Sydney. That was fantastic. And there's so much to learn about mushrooms. Our next speaker is Dr. John Flynn, who's a neuroscience professor at Rice University who specializes in computational neuroscience. Dr. Flynn will be talking about making AIs hallucinate, theories on how the brain processes information during psychedelic experiences, and how both help us understand how the brain usually works. John?
All right, can you hear me well? Cool. So I am a computational neuroscientist, at least in my spare time. And I approach some of these questions a little bit differently than a normal biologist would. A normal biologist might ask, when you ask the question, what does psychoactive drugs do? They might say, okay, how does the molecule fit into a receptor and then affect the cell? Alternatively, they can say how a molecule, how it causes a cascading reaction that increases or decreases various hormones or neurotransmitters in the brain. They could also talk about things at a systems level, which is how groups of neurons work together to be able to try to create phenomena of the mind. I treat the brain a little more like a computer than a chemical soup. And this isn't bad. This is a different way of approaching it, but all of them have benefits, all of them have drawbacks. The brain's incredibly complex. You need all types to try to figure it out. To give a sort of analogy, what computational neuroscience does with the brain is looking at traffic patterns. So I can look at traffic patterns in, say, New York City without understanding how an engine works. I don't really care how the engine works. I mean, I do. I think it's absolutely fascinating. Same thing with neurons and whatnot, but that's just not really it's not really relevant if I'm trying to say, when a highway will be congested at what time. On the other hand, sometimes there are biological phenomena that happen that end up creating larger scale effects that I might be interested in. So if you happen to have Firestone tires in our traffic analogy that end up, being inclined to blow up, that might cause traffic jams. Just like if you toss a psychoactive molecule into the brain, that might do something really strange to the information processing that the brain does. So that's why, somewhat interested in psychedelic drugs, and we'll get back to how they play into it. Let me first talk a little bit about sort of the abstraction of neurons. Neurons are what make up the brain. They're incredibly complicated. They have all sorts of physiological features down to the nitty-gritty molecular level that all work together to make the entire brain work. But you can somewhat summarize up a neuron and to roughly approximate it by saying when this one fires, it sends a signal down to talk to this one down here. And it might, if it has a strong enough connection, cause this one to fire and send a signal further. And we can talk about the connections between neurons. So if this neuron fires a couple times and it also causes this one down here, let me go ahead and get a laser pointer so that people can see what I'm talking about. If we get a signal that sends a signal down here to cause this one to fire every time this one fires, that's a strong connection. That means that that these basically have a one-to-one connection. If it takes a lot of firing from this one to make this one fire, that's a medium strength connection. And if this one fires a lot, and it barely makes this one fire at all. It's a weak connection. And we call this the weight between neurons, how strong that connection is. And you can build these things called neural networks that you may have heard about. They're kind of all the rage in computer science right now, and they're somewhat based on how the brain works. Not entirely, but they're related. And basically it is that rather than having complicated neurons that have cells and nucleuses, or cell bodies, nucleuses, dendrites, myelin sheaths, all that, they're circles. And the only thing we care about is how much this circle affects this circle whenever it fires and sends something forward. And this is called, this particular example, where you have a group here sending signals back and forward to a group here, sending signals forward to a group here, sending signals forward. One term for it is a convolutional neural network, or a CNN. And this is just a complicated name for something that's used quite often. The quintessential example of where this is used in your everyday life is if you ask Google, is a picture a puppy? Google uses these networks and trains them to be able to say, which one of these is a puppy and which one is not a puppy. And the way that that works is very similar to, say, or somewhat similar to, a child. A child babbles, and you give them a picture and you say, is this a puppy? No, it's a cat. No, that's not quite right. This is a puppy. Try again. Is it a puppy? And just like a child might say, puppy detected. That might be a very odd child. You would have, if the neural network had a neuron at the end that was the puppy detector, go off and say puppy detected, you would praise the network. Mathematically, it's a little more complex, but you say good convolutional neural network, and the weights in between these fake neurons change. And it learns. Likewise, if we give it a picture of Robocop, and it ends up going through all this process, these neurons talk to these neurons, talk to these neurons, talk to these neurons, and it still says puppy detected at the end, you say bad convolutional neural network, and you punish it. This is probably where HAL kind of went bad in 2001 Space Odyssey, but this does give us really, really good results. If anyone remembers the internet back in the 2000s and the early 2000s, this was not feasible. You could not say puppy pictures and get 10 billion puppy pictures very easily, whereas now it's trivial, and it's largely thanks to these convolutional neural networks. This is pretty good. The only problem with these, and just like the real brain, is that we don't know what the individual neurons do. And we'd like to know what the individual neurons do. We know that there's a puppy detector kind of thing at the end that will say yes or no, there was a puppy detected. But we'd like to know if one of them is more for eyes or ears or noses, or maybe this one's for like texture for the fur, and it kind of turns on whenever you have something fluffy at the beginning. And there's a bunch of ways to kind of do this, and roughly you find that, frankly, it actually ends up looking like the human brain if you can, for some of the techniques that we've had to try to figure out how these different layers work. Some of them, like the first layer, ends up being focused on lines and colors. We see the edge of the puppy here, the edge of the ear. That feeds into maybe an ear detector down here, maybe, and then we have more shapes. These shapes become bigger things that are features. And then we get to the objects. But it's still kind of in its infancy of trying to figure out, for us to try to figure out what's going on inside this network. It's a black box. We don't know. So there was an attempt to try to figure out, to use a new technique to try to figure out where the nose of the puppy was in this network. Where is the nose neuron, if that makes sense. And so a group of researchers in 2015 decided to see what would happen if they ran the network backwards. And to all the computer scientists, all three computer scientists in our audience, I apologize for how much I'm going to elide some of the details in this. I'm sorry. but where they basically ran the image backwards to have put the picture in at the end and marked had the neurons mark the picture as it was going back and say like, okay, if you activate with this area, make a small change to the picture and show us what you're affecting, what you are picking up on the picture. And this is like, hopefully they want to see like, you know, if we could see where if this one marks the nose a lot, we know that, hey, we have the nose detected here. And so they found something interesting and they released the algorithm online. And some of you may have seen this at some point. But I did this afternoon with a picture of the Sally Portlet rice. This is where the graduates go whenever they are going, you know, a normal year, non-COVID. They walk through here, after they get all their diplomas, they play the music, yada, yada, yada. And if you apply this particular algorithm, you go from the Sally Portal at Rice to the Sally Portal to Oz, and it becomes a little psychedelic, and it's kind of neat. This was, again, popular for a couple of years. I suspect many of you have seen it, but it's kind of has some interesting implications for neuroscience. So just to give you some other examples, this was a picture of Shiba Inu that was put through an animal detection network, or reversed through the animal detection network several thousand times. And you can see the main dog shape here, those are the original eyes. And we have an abomination before God or something like this growing out of the puppy. Here, a donkey looking thing here from space. We have a example of an English riverside housing that has been infested with eyes. And we have various horrifying things with faces inside faces inside faces. We also have, if applied to an extreme level, you know, some interesting geometric patterns that seem to pop up to a large extent. And to people who were, you know, the alternative rock fans in the early 2000s, and perhaps like Tool as much as I did, this starts looking a little familiar. And you might be, if you were familiar with the band Tool, you also might be familiar that they did a lot of DMT, which is a hallucinogen. And this is kind of the result of some of their artwork, which, you know, not perfect, but seems relatively similar. It's not enough to base a scientific principle on, but it's worth some introspection. So, what's going on here? Well, first off, you do not see the world as it is. I'm not trying to be deep here. I'm not trying to be mystical. You just literally do not see the world as it actually is. I think this is one of my favorite illusions of all time. I promise it relates. So I usually have a class when I'm doing this, but so I'm just going to assume I'm getting answers from the audience. But I want you to look at this is called the sofa cushion illusion, because they have sofa cushion, it looks like a sofa cushion on top of another sofa cushion, kind of like pressed inward like that. And the top one looks dimmer than the bottom one. And I know you all are clever. I know that you all have figured out that yes, these are actually the same color. And if you want to prove it, go ahead and raise your finger up in front of your eyes and put your finger right where the horizon is, and take out the sort of white and dark space between. And it should pop fairly quickly that, oh my God, these are definitely the same color. And the moment you take your finger away, you will, Ariel, do you see that? Okay, cool, just making sure. The moment you take your finger away, even though you know that they're the same color, it pops back in to being the distinctive colors. And this is because your brain is trying to help you predict the world. This is called predictive processing. And one of the ideas of what the brain does is to try to help you understand the world, because your eyes are moving around all the time. Even though you think you see, a single image of the room or the screen in front of you, you only see a tiny bit of it, and your brain stitches it all together for you. And it helps you in stitching this together by predicting what you should see, because you expect certain things in the world. And in this particular illusion, you are coming in with the expectation that you are seeing a horizon. And you are outside, and the sun is shining down on top of the sofa cushion on the top, and there's a shadow being cast on the sofa cushion on the bottom. So your brain automatically decides, well, look, I can tell from the raw data, they're the same. However, I know that if they look the same when one is one is in shadow and one is in light, they have to be different colors, and I'm going to adjust for that. That's why we get this strong illusion, and this is called top-down processing. This is in comparison to bottom-up processing, where you know that, or where you're looking at the raw information and trying to figure out what the actual color is. Top-down processing is trying to figure out, what you should interpret from the color, and much of your conscious experience is top-down processing. And so we have here a couple other quick illustrations of top-down processing. The first one, I love Paris in the springtime. And again, this would be great with an actual audience participation. But how many of you saw the twice in this? I love Paris in the springtime. So others of you, we also have here the cat. Fairly straightforward. It says the cat. What else could it possibly say? Of course, the H and the A are exactly the same. Not that that bothered you while you were reading it. And this is one of those really subtle things that, like, this is really weird when you think about it, that you have absolutely no compunction with making that an H in one case and an A in the other. But that's because it's the H in the first case is surrounded by a T and an E, and the A in the second case is surrounded by a C and a T. And you know ahead of time, you expect the world to be in a certain way, so you automatically adjust your perceptions to better process the world. And this is good. You really want to do this. You don't want to leave your house in the morning when the sun is at a certain angle, come back later in the evening when the sun's at a different angle and say, where did my house go? Because it's a different color. You want to be able to make these adjustments. Finally, if you're wondering what the heck this thing is, it's a cow. And you have the ear here, the outside of the face here, eye here, nose down here. Need to adjust this. And nose around here, back over here. And now that you see it, if you see it, some people have a hard time with it, you can't unsee it. And that's another example of where top-down processing takes the lead. And so that's kind of what's going on with this neural network thing. We have here a image going backwards. Again, I apologize, computer scientists. Going backwards through this being marked up and saying that, okay, I'm interested in eyes. And so I'm going to mark everything that looks like an eye to me whenever I go through. And it's just gonna be a tiny mark. And it goes through, and then you run it through again, and again, and again, and again. And if you run it through, you start with this image, you run it through 10 times, you get this. A little trippy, not that bad. And you start seeing, just like people see, clouds that have faces, or the eyes. Some of the windows seem a little bit like faces. This is what you do when you're a kid, before we had smartphones to distract us all the time. Is that you would look up at the clouds and you would play this game where you would see things in it. We're just taking this and ramping it up and doing it mathematically. If we do this another 10 rounds through, it looks like this. You're starting to see like this demonic dog over here. This network really likes dogs, by the way. We crank it up a level notch. We've given Google LSD, that sucks. And then you're having a bad trip in this particular one. So you keep running it through. And essentially what you're doing every time is you're increasing the amount of top-down processing. You're running the system backwards, where the things at the top are controlling the image at the bottom. This is actually, like I said, I mentioned earlier, this is somewhat how the brain works. That we have a fake neural network that's running at Google. It's not Skynet. Don't worry about it. But it starts by looking at images with lines and colors and small features at the beginning. It groups them into shapes. It groups them into features. It groups them into objects and says whether or not they are a certain type of thing. And one of the things that this aligns in biology, that biology has that this doesn't, at least not straightforwardly, is a feedback network. And biology has built-in feedbacks to combine top-down processing and bottom-up processing to help that what I was talking about earlier. You have these predictive processing networks in your brain that help you see correctly, or quote unquote correctly, despite the fact that these two colors are the same. It lets you perceive the world as it should be, at least your internal model says. So we do have these feedback networks, and this somewhat is similar to what we talked about with sending the image back through it several times. That's a type of extra feedback. And we can actually see this in the brain. I'm not going to get too much into detail in this model, but one way of looking at a trip, if you had a psychedelic experience or something like that, is that you have a sensory input, you have a strong internal model of what the world should be like. It affects your perception, your lower level perception of the world, and then that is transmitted to your top-down model and says, essentially, like you start off with a circle in the background. It says, I think that might be an eye. Okay, maybe it's eye-shaped-ish. No, I really think that might be an eye. Okay, fine, it's kind of eye-ish, and like okay, it says that. Definitely this is an eye from the new piece of data, disagrees, and they start creating, shapes out of random objects. This is where you go from seeing, a cloud that, yeah, it's kind of a face, to if you crank up the top-down network a little bit in these neural networks or the feedback loops, you start, seeing Mufasa in the clouds or something like that. There are some really cool papers, and I'm not going to get too much into this figure. There's some really cool research right now going on trying to use some of these convolutional neural networks and comparing them to real life neural networks that we know, and looking at, okay, if we shut down the network at this point, does it give us a DMT-like trip? Does that help explain why we see faces like, or someone might see faces like this under the influence of different drugs? If you get a concordance with people who have taken the drugs and saying that, yeah, this is kind of what I see, then you might be able to use that to say, look closer in the brain and say, okay, it might be these neurons that are causing this specific effect. And so by using this, these sort of convolutional networks, you can kind of understand where the brain, quote unquote, breaks during a trip. And this, in the end, might help us understand how the brain works when it's not breaking, if one considers that breaking. So I think this is really cool research. And I think this is a really cool way to apply math and computers to help us understand how the brain works and improve our general understanding of the world. That's all I really have to say.
Awesome. Thank you so much, Sean. That was very fascinating, and puppies are always a good example to use. Now it's time for our Q&A segment where we'll answer your burning psychedelical questions. If you ask a good question, we'll put your name into our digital hat for a prize drawing, which we'll announce in the Facebook event page later tonight. So you could win your very own Taste of Science tumbler cup. So our first question is more of a medicine question. Does anyone know if drugs, marijuana in particular, interfere with medications?
Spencer, are you going to take that one?
Yeah, for a second, I wasn't sure if we were supposed to answer. I mean, certainly they can, drugs can affect metabolic pathways, whether they're the cytochrome P450 system or whatever enzymes are used. They can affect the absorption, by how you're using marijuana. They can affect receptors. A lot of the drugs we use, well, a lot of drugs that are used recreationally, again, I've never done one, started out as experimental drugs to study various body systems. I mean, look at our synthetic cannabinoid epidemic. They all started out, back in the 60s when John Huffman from Clemson was trying to study the endocannabinoid receptors, for potential treatments for MS and other immune or possibly immune conditions. Yeah, so they all have multiple effects. And if you use certain ones, then they can enhance or antagonize the effects of others. They can prevent the absorption, enhance the elimination. They can have effects on all sorts of kinetics and dynamics. For example, certain opioids are influenced by certain antidepressants. Tylenol with codeine is essentially Tylenol with a stomach ache if you're on certain SSRIs because you can't activate the codeine to its morphine. So that's a very long answer for a simple question. Yes, marijuana can affect, but you'd have to ask a more specific question to get a more specific answer.
Would it be okay if I added something onto that real quick?
Please, yeah.
I think a good mention for sure is CBD, and this has actually been implicated in a number of poisoning events. CBD is a relatively potent inhibitor of certain cytochrome enzymes and has been known to potentiate a number of drugs to potentially toxic levels. So it's important to keep in mind that, even though CBD, for example, isn't psychoactive like THC is, avoiding mixtures is probably always going to be a good idea there. You should keep in mind that it may potentiate other compounds.
Awesome. Thank y'all so much. I have another question for Sydney. What does stains green mean for someone unfamiliar with magic mushrooms?
Yeah, I'm happy to answer that. My video cut out, by the way. I'm not sure what happened there, but my camera won't restart. So there are a number of color changes that can actually happen while you are handling or investigating mushrooms. Now, like psilocybe cubensis is a really great example of that. It stains brilliantly blue when you damage it. So like if you pick this mushroom, you'll often get blue marks appearing where you touched it. So you'll have like finger marks appearing where you picked the mushroom. If you chop it or crush it or damage it, you'll get this blue color. And that's actually from oxidation and production of this blue pigment from psilocin. So that's actually from the compounds themselves joining together. Now with Gymnopilus, Gymnopilus luteifolius is like the common psilocybin containing Gymnopilus, different species, different group than psilocybe. That one stains blue, but because the mushroom is orangish reddish, it comes out as more of a green color. But mushrooms can stain yellow, red, like black. I mean, it's all over the place. There's a lot of color changing mushrooms out there.
Awesome. Thank you so much.
Yeah.
For John, someone asked, what happens as the number of neurons decreases? Is there a threshold under which you can't hallucinate? And can we make leeches hallucinate? I don't know I don't have any context for that.
I see the rest of the question in the Zoom chat. That's a really interesting question. So in general, to start off with the first part, if you were to randomly remove neurons from most of these from the models that were handled, usually they're fairly robust. It's kind of like if you randomly remove a few neurons from your own head, you don't just suddenly lose massive amounts of ability. There's a lot of redundancy built into them. There are some attempts to make very minimalistic neural networks to be able to do things, and I think but I don't think that's been used for deep dream. As for whether or not you would a leech be able to hallucinate, with the idea that that perhaps this brain is so simple that it doesn't have the correct feedback loops. I actually think that. I actually think that maybe you're right. Maybe the questioner's right. That with no feedback networks, I'm not sure that it would have hallucinations in the same sense because it doesn't have an internal model or with fewer feedback networks, it doesn't have as much of an internal model of the external world. So it still might not see the world properly, but it wouldn't see like leech eyes or the shape of other leeches in the environment or other things like that, because it's not complicated enough to store that information in its head. And so, on the other hand, whenever you're trying to speculate it on the subjective experience of other entities, particularly non-human ones, it gets really specious real quick. So.
Fascinating, thank you. And Spencer, I have one whimsical question and one more scientific question for you. Someone in the chat asked what your cat's name is.
So this is Desi Danger Cat, and he and his siblings, Lucy Goosey, Ethyl Chloride, and Fred Rogers, they're all turning 2 on Friday. They're obviously, quadrupeds, and he is 15 pounds, and the other's like 8 and 9 pounds. He is our defensive lineman. So I'm assuming that was the whimsical one, maybe not?
Yes. And then we also had someone asked, is there any drug that the chill out, metaphorically and figuratively method does not help you recover from, and anything that requires more active intervention?
I mean, so with Jimson weed and other antimuscarinic toxins, we'll take a step back. I made a comment about benzodiazepines being our best friends in the emergency departments, and many emergency physicians have joked that benzos should be readily available to everyone in the hospital. They should be aerosolized so that they're affecting patients, staff, and visitors. Benzos are great for many things. When you're agitated and hyperthermic from some sort of sympathomimetic, when you're, serotonergic, they're wonderful, but they don't do anything for antimuscarinic delirium. Multiple studies have shown that they may tire you out a little bit, but you're still delirious. But we still want to calm them down because, people die from one of several ways when they're under the influence. They can be injured, of course. Beyond injury, a lot of them get volume depleted. There's a lot of heat dissipation and volume loss, either directly from vomiting or diarrhea or indirectly from sweating, and insensible loss from, breathing harder. So we want to fluid resuscitate, and that will also cool them down. We want to control the temperature too, because if they get hyperthermic, they'll, disrupt multiple systems. They'll literally and figuratively fry their brains. They'll have status seizures. They won't be able to have, maintain their normal homeostasis. So it's important to literally and figuratively chill these people out. Sometimes it'll keep them from hurting themselves. Sometimes it'll actually fix the problem. Someone comes in on speed, we give them some benzos, they calm down and they can, be lucid again. Others, we have to do something that specifically addresses the delirium. And that would be, again, antimuscarinic toxicity, for example, we give them physostigmine. So, and I saw another question later on, this is sort of related. Sometimes we don't know frequently we don't know what's going on. People have this impression that we do this great drug testing and we know exactly what someone's on when he or she comes in. Most toxicologists refer to the UDS, which most people think is urine drug screen. We refer to it as the useless drug screen because it's fraught with false positives, false negatives. Not to mention, it doesn't catch the majority of substances on which someone can overdose or the majority of substances that someone can abuse. So we often don't know what's going on. So we focus on basics. We, make sure they have a protected airway. We oxygenate them. We ventilate them. We make sure they have enough fluid, but that the blood pressure doesn't get too high or too low, and then we cool them off. And if they're hypothermic, then we warm them up. And then, we look for lab tests or EKGs or physical exam findings that point to a specific sort of toxidrome or specific substance, and then we give them antidotal therapy if one exists. But yeah, oftentimes we don't know what's going on. We just have to do the basics, chill them out, literally and figuratively, and just keep them alive until the drug is gone or the effects have resolved.
Thank you. For whomever would most like to answer, what is horizontal gene transfer? I know someone mentioned that earlier. I personally can't remember who it was.
That was me. So horizontal gene transfer is more or less what it sounds like. It is the cross-species, well, general context, the cross-species transfer of a gene or group of genes, viable genetic material. So in this context, you basically have a gene cluster. You have this group of genes responsible for coding most of the steps for producing psilocybin and psilocin. So basically, this is like the genetic code that enables an organism to produce these drugs. Now, the horizontal gene transfer is going on where if you have a scenario where these two fungi are cohabitating, two different species, horizontal gene transfer would actually be somehow, mechanism is currently unknown, somehow that gene transfer gets transferred to a different species that previously didn't have it. Now, a lot of like genetic modification methods where you transform organisms to produce new proteins or you give them new genes. This is pretty similar to that, except in a natural context. You know, we can alter like glowfish, these silverfish, to produce like crazy neon colors by inserting certain genes. The same thing is going on here naturally, but with the psilocybin gene cluster. And so the really significant thing is the mechanism is unclear. We've never observed it happening, but because this gene cluster is mostly identical between all of these different species, the evidence that it's been transferred horizontally is very strong. And it's such a long and complex gene cluster, it is very unlikely that it could have developed independently so many times. And you know, in some contexts, horizontal gene transfer happens all the time. Like a really common one is where you get parasites will have horizontal gene transfer with their hosts or vice versa. You usually require some form of closeness, some relationship that puts them physically close together and increases the likelihood. But again, we have no idea how it might have happened here. And in a lot of cases, the species that are exchanging these genes don't live in the same places. So there's some interesting vector. There's something going on, but we don't know what.
Fascinating. Thank you. Our next question. There's still a lot of stigma around illicit drugs, and there are still people in prison for marijuana charges in states that it's legal, which is another more social than scientific issue. But there's still a lot of stigma, so much so that policymakers don't want to suggest legalizing their use for medicinal purposes in some cases. So how are scientists able to go about doing research with drugs like these? Is there a lot of red tape to go through? How does that work?
Yeah, it often doesn't. I mean, so the simple answer is that if you want to have a stable scientific career, you don't research these things. There are a few exceptions. There are some people who have made their names in it. But the amount of red tape to go through to get to test anything with a Schedule 1, Schedule 2 drug is just insane, which is pretty terrible because it is like, for example, cannabinoids, THC, marijuana. These things, the cannabinoid system is one of the largest receptor systems in the brain. Like, you have a huge amount of neuron to neuron communication that's driven by some type of cannabinoids. And we don't really know anything about it because no one wants to deal with that. So I like it. If for no other reason, I'm looking forward to this wave of legalization that's happening about that. Maybe we can start studying this stuff.
Thank you, John. And I think, Spencer, did you have something to say as well? I'm so sorry, you're muted.
I was going to say I have nothing useful to say. The question, how do you research on it? I mean, there are exceptions. You know, there are certain drugs that have different schedules for different indications. For example, GHB, it's Schedule 1 for most things, but Schedule 3 for narcolepsy. There are ways, you know, relatively simple ways of getting permission to do specific types of research, but that's not what I do, so I can't answer it any more specifically than that. But there are ways. I mean, cocaine has therapeutic applications. You know, it's part of TAC, one of the local anesthetic combinations we use. So there are ways to do it, but, you know, like John said, it's a fine line. You know, it's probably safer for your career to do something that's a little more wholesome, for lack of a better word.
Thank you. And for whoever would like to answer this one, does the method of taking a drug change the effect the drug would have on neurotransmitters or clinical features?
So let me start with that. I think John's probably going to answer as well. You know, pharmacokinetics are loosely described as what the body does to the drug, and pharmacodynamics are what the drug does to the body. The route is part of pharmacokinetics, but ultimately it shouldn't really affect the pharmacodynamics. Now it'll affect the dose, and of course the dose determines a lot of things, but it doesn't change its inherent mechanisms of action. It doesn't change what it does, but it'll, you know, either happen faster or slower depending on the route, and that can have a larger or slower dose, you know, a larger dose or a smaller dose. So it shouldn't change what it does, but it should change when it does it.
And as a quick addition to that too, there is the like prodrug or metabolite dynamic to consider as well. Like many drugs have to go through an initial metabolic step in order to be available. Like, you know, the ibotenic acid mucimol and psilocybin psilocin are both examples of that. If you were to, well, now location of these enzymes notwithstanding, this is an example, like ibotenic acid and mucimol. When you take ibotenic acid orally, it goes through metabolism in the liver on first pass. So after you eat it, before it enters your bloodstream, well, before it enters main circulation where it can access your brain, it gets hit with these enzymes that convert it to mucimol. Now, if you were to inject that ibotenic acid or go a mucosal route, you know, like snorting is an example of a mucosal route. No one's out there snorting ibotenic acid, but just in terms of routes here, if you skipped the liver by smoking, injecting, or mucosal absorption, you may not get that first pass. You may not metabolize it. And so instead of getting mucimol that you would from eating it, you might just get all ibotenic acid. And, you know, same with psilocybin, you may get all psilocybin instead of psilocin. But, you know, it's really context specific. But if you're just talking about a drug, like, you know, if you're only talking about psilocin, certainly route may affect speed of absorption and the dose curve over time, but, you know, ultimately the neurotransmitter dynamics are going to be pretty similar.
Right. Yeah. Sydney mentioned something really important. You know, even with normal therapeutic drugs, the route really does make a difference. That's why, for example, we don't take insulin orally, why we have to inject ourselves, because it's not absorbed. Or, you know, when we use sublingual nitroglycerin when someone's having, you know, possibly a heart attack, it's not absorbed very well when taken orally, but if it's absorbed sublingually. So yeah, you know, kinetics include absorption, distribution, metabolism, excretion. If one or more of those is affected, whether it's first pass or absorption or whatever, then they won't get the effect. But like I said before, the dynamics are the same. It just depends on what's ultimately doing those dynamics.
One thing to add to that is more on the behavioral side, is that the addictiveness of something is almost entirely proportional to how fast it hits. So if you like marijuana, not terribly addictive, because whenever you do a marijuana cigarette, it isn't going to, it takes a little bit of time for it to hit. Whereas if you were to take a nicotine cigarette, it hits like that due to a variety of factors. But if you could delay the nicotine hit, you wouldn't have as much of an issue. This is one of the reasons why, say, for example, Adderall often has extended release. It's amphetamines. So it has a high addictiveness potential. But if you can slow the release over the day, as opposed to like, boom, you largely reduce its addictive potential.
Thank you all so much. What role do cultural traditions play in research on recreational drugs, if anyone has any experience with that? For example, ayahuasca has some complicated rituals around it from cultures in the Amazon basin, from when to pick it, how to process it, how it's consumed. Do these get included in this type of research, or is the cultural aspect of it more just focused on in like an anthropological setting more so than this type of setting?
I can briefly comment from the perspective of sort of top-down versus bottom-up processing. If one reads any books from the 70s, set and setting, so mindset and the setting you're in ends up changing the trip a lot, and I think that makes sense within sort of the computational model that I've suggested today, and has been suggested in other papers. If you're inclined to be in a certain mindset to expect things, then it's going to change your perspective of what comes after. And for something that affects your top-down processing hugely, that's going to be really important. I don't know of anybody who's actively researching this type of thing. But it would strike me as potentially something that would be useful.
Awesome, thank you. And someone asked, why do plants or fungi create psychoactive chemicals? Is it beneficial to them in some way?
Well, I mean, in a big way, that ties into kind of what I was talking about at the end of my little slideshow. Is the role of some of these things is something that in many cases needs to be researched. For many of the compounds in fungi, it's pretty strongly suggested, theorized that they're often anti-predatory, sometimes even anti-fungal. These compounds usually have a role in defense, and that's often true for a lot of secondary metabolites in general. And when you're talking about secondary metabolites, these are compounds produced that are not directly essential to like basic maintenance of life, growth, and development. They're kind of extra. They often serve accessory roles that facilitate the lifestyle or ecological role of a species. But in plants, there is very well established anti-predatory roles. It's much more well researched, and like nicotine's a great example, potent insecticide that is directly observable to play that role in the wild. Like it's pretty well established for many plant compounds. But then when you get into things like DMT produced in plants, potent psychoactive drug for humans, maybe most mammals, but the role that plays in a plant is not well established. In many cases, it's at such low levels, it can't play any kind of anti-predator role. And so the actual function in many cases is not well understood. But DMT, like psilocybin, is so widely distributed among plants, it's likely to play an important role. We just don't necessarily know. But we have to kind of examine these on a case-by-case basis. Really, the metabolite diversity out there is insane, and there's so many different possible functions. But certainly, I would suggest looking into reading on plants in regards to some of the psychoactive drugs, just because there's a longer history of research. With fungi, like I mentioned, a lot of these things aren't well established.
Thank you. It makes a lot of sense that it would be an anti-predatory sort of thing. Someone asked, what's the actual potential of marijuana as a medication? Everyone's selling CBD products for everything from depression to cancer. Is there validity in this, or validity in some of this and not other aspects?
We know certainly there are some medicinal benefits. You know, various derivatives have been very effective in treating a variety of nausea, especially refractory nausea. There are some components that have provide good topical analgesia. So yeah, I mean, you guys can add on to that, but there's certainly some components that have proven medical value.
Definitely. I mean, I'm certainly biased. I think most of us here are biased just in that, you know, I really like to see scientific support. I really like to see clinical, or at least kind of any data suggesting support for some of these proposed medical roles. And certainly what I will say for CBD, at least, is many of the things it's sold for are not strongly scientifically supported. And, you know, anecdotes are important, but I'd be cautious, especially if it's being used to sell something. Certainly, I am a little cautious of the almost like panacea way that CBD is sold, and, you know, THC to a lesser degree, but CBD is a little more complex because it's not immediately psychoactive. And I think it's easier to say it's doing something on the sidelines, solving problems. But, you know, anecdotal support for some things like joint pain, arthritis, anti-inflammatory effects, specifically in the context of joint pain and soreness, the weight of anecdotal evidence is pretty enormous. I'd suspect there to be validity in some of those applications, but again, really, be cautious. And I'd highly recommend, if you go to Google Scholar and just search, like, often the keywords cannabidiol, that's the long name of CBD, cannabidiol, or CBD, and then literally just whatever illness you're interested in. You can often find what literature is available out there. I'd always recommend people do that for almost any drug they take.
Thank you. And on that note, I know we've spoken about the difficulties of red tape with some of these things, but does anyone have anything to add to that on if other recreational drugs have underutilized medicinal properties such as ecstasy, psilocybin, LSD?
Well, red tape has certainly gotten in the way of, I mean, really just effective research of most of these. I mean, you know, there's just not, like, it's kind of hard to say exactly how much medical potential a lot of these compounds have when, like, we really just don't, we haven't looked hard enough for many things. Like, I don't know, the efficacy of microdosing, LSD and psilocybin both are very understudied, yet it is one of the most popular uses of these drugs by far. With psilocybin especially, it's getting more and more popular, and we really need studies on it. And a lot of that red tape is definitely getting in the way of that research. You know, there is an LSD study published, I don't know how many months ago, but they used a really weird format for their paper due to this red tape dynamic. They basically did a mobile study. They had users who identified as these were people who were already interested in or using LSD therapeutically. They basically reached out to get their participants online. These people bought their own supply of LSD. And then these researchers sent out capsules, blinded capsules, and had these people run blinded trials on themselves for microdosing. And the design is quite interesting. And then, you know, they gathered actual attempts at quantifiable data with surveys, basically. And the design is quite interesting. The concept is really interesting. But, you know, unfortunately, the methodology leaves quite a lot to be desired. You know, even simply something like when you're buying LSD off the street, you simply don't know what's there. And so if you can't control dosage for an attempt at a trial on seeing what doses do to people, there are certainly flaws, but it's an interesting example of how researchers are really like trying as hard as they can to get data on these things. I mean, certainly the popularity is outpacing the data available.
Thank you.
I'd also just quickly like to add, there's some really cool stuff with psilocybin treating depression that people are working on, as well as things like ecstasy as an pathogen and an aid for some types of therapeutics that it might make someone more empathic to the researcher, which could, or not researcher, sorry, the psychiatrist, and that would potentially have some grand benefits, or to due to couples therapy or something. I don't have. I think those are a huge amount of promise, but you know, we don't want to oversell it in the same way that, you know, it's not a panacea. It's not like the government doesn't want you to know. It just is an unfeeling thing that that crushes research accidentally.
And that sort of leads into another question, which is, does anyone have a particular take on stoned ape theory? The idea that proto-human ape ancestors eating mushrooms led to opening of the mind, or even brain size increases in some aspects. I have seen a few articles about opening the mind recently, but I don't know the validity of that.
So what I'd say is that unfortunately McKenna, that it is his theory, Terence McKenna, one of his most popular legacies, I guess. He was not a mycologist nor really anthropologist in the classical sense. The theory is really mostly just, like, I don't know. I don't want to completely dismiss it and just say like he was making some stuff up and it doesn't make any sense. It's an interesting thought. You know, when it comes down to the details, things like the occurrence of psilocybe aligning with human development, you know, the speed at which some of these developments in like cranium size happen, none of it really lines up. Like it is unlikely to have actually happened, but I don't think we can necessarily discount the role that psychedelics might have had in prehistory. I certainly think that we can't discount some use, but the overall proposal that an ape-like pre-humans ate psilocybin, became smarter and turned into modern humans is not based in science. But certainly, I think it's historical use of psilocybin and other serotonergics and just psychedelics in general is something we should look into. I mean, just look at how a drug like ethanol has impacted societies for so long. Like, I certainly don't think we could discount, like if a drug manages to change society, even economically, by building a society or contributing to it, you could argue that it contributed to the development of humanity. But no, they did not make our brains bigger after eating them.
Thank you so much. Does anyone have anything else to add to that before I move to the next question? Okay, we'll try to get to as many questions as we can, but we'll probably wrap it up relatively soon here. The next question is, can the use of marijuana or other common recreational drugs have an effect on vaccines, a COVID vaccine specifically? Do we have enough research to determine this?
I don't know if we have any research on that.
No, none.
A priori, I mean, so I'm going to spitball here. A priori, I'm not sure, unless you actively took it at the time of your dose and you were interfering with your immune system by putting yourself in one of these like these hyper-aroused states that Dr. Green was talking about. I have a hard time seeing how it would interfere. I mean, the body is complex and you could totally be wrong, but a priori, I would not bet on it.
Exactly. They shouldn't have any effect, you know, any immunomodulatory effect that would prevent the vaccine from doing its job, but yeah, I can't think of anything.
Yeah, I mean, with the mRNA vaccines, at least, the way they function is just so integral to basic cell function. Like, you know, they work on such an integral level. I mean, I don't think even many immunomodulatory drugs would impact it. But again, I don't think there is even a hint of research on that.
Thank you all. Is it true that antipsychotics can block the psychedelic effects of psilocybin mushrooms? And how would this work if this is true?
I mean, I have no practical experience with this treating anyone, but it would make sense knowing that, you know, these psychedelic mushrooms mediate some of their effects, or the majority of their effects, through certain serotonin receptors, and certain, you know, psychiatric medications antagonize those receptors, then yeah, you wouldn't be able to get the effect. So it all makes sense in theory. I don't know how much that's been studied in a formal setting, but it certainly makes sense mechanistically.
So I can't speak to antipsychotics, but with selective serotonin reuptake inhibitors, so like SSRIs, there is a viable mechanism that's been proposed, a viable mechanism where these drugs can interact. And there's also a long history of anecdotal evidence for partial or total inhibition of activity. So we've certainly seen things like SSRIs interact with MDMA, LSD, and psilocybin, and the theoretical pharmacodynamics for this at the receptors add up. So it hasn't been confirmed, but it is pretty likely and also often observed that SSRIs can completely inhibit or modulate the effects of psilocybin, LSD, and MDMA.
Thank you. And how does a person's weight, gender, other physical characteristics influence the effects of different types of drugs?
Oh, well, obviously, someone who has a lower body weight, they're getting a higher milligram per kilogram dose. You know, if you have a higher percentage of body fat and it's a fat soluble drug, that'll absorb, you know, that'll distribute into more tissue. I mean, there's a whole bunch of factors. And then of course, there's tolerance too. So, I mean, there's so many factors that determine the effect. You know, it's hard to predict how someone's gonna respond, but certainly a larger dose in a smaller person, or, you know, again, how much distribution will affect, you know, the magnitude and the rapidity of the effect.
There was a paper published on body weight adjusted doses of psilocybin or psilocin, where it was either psilocybin or psilocin, but they gave 25 milligrams to a wide range of people, and unlike prior studies which had weight adjusted doses, this study gave 25 milligrams to everybody and then rated their experience and then compared that data back to body weight, BMI, etc. And I don't recall the extremes, the max and min of their weight range, but they didn't see any observable changes in just the standard realm of BMI. They did not, at least with this dose of 25 milligrams, which is a strong dose,
Yeah.
they didn't see observable changes.
Very interesting. We'll do one or two questions and then we'll wrap it up here. Someone mentioned that you could add things to benzene rings to mix and match effects. So if a drug company wanted to, and were allowed to, more importantly, do you think it would be feasible for them to optimize a recreational drug while maintaining safety, or would we not exactly have an idea of how to go about that?
Well, they already do. Yeah, I mean, to an incredible degree. I mean, like Spencer mentioned with synthetic cannabinoids, I mean, in many cases, one of the first things a pharmaceutical company will do when they encounter a new drug, essentially they treat it as a backbone and then explore sometimes hundreds and hundreds of derivatives or altered forms, or sometimes it goes the other way where they identify a receptor that's important to a disease or disease state. And then they will kind of back engineer and produce hundreds of compounds in an attempt to find something with affinity to that receptor. So it's certainly something that's already going on. And, you know, there are some exceptions, but I mean, there's not, as far as I know, there hasn't been too much in the way of producing a lot of these novel compounds historically. You know, there is the Federal Analogs Act, where if a drug can be considered an analog of a scheduled drug, it is automatically on the same schedule. But certainly, like what you're talking about is basically substitutions, substitutions or additions. And I mean, like just for example, with like cannabinoids, they basically tossed hydroxyl groups, lengthened the chains. They've like really just altered almost every aspect of that molecule in investigated activities. And the vast majority of these have never been tried on humans. But recently we've been doing things like running them through synthetic, or not synthetic, in silico receptors, like basically modeling how these things might affect parts of the human brain, mouse studies, etc. So really the hard part isn't necessarily making a lot of these, it's just that it takes billions to actually get them to test on people. That's the hard part, is getting people to try it, not necessarily producing the diversity.
Yeah, I mean, we have hundreds of different synthetic cannabinoids that differ slightly. And then all the synthetic cathinones, you know, I showed you the structure of amphetamine. Well, COT, you know, has cathinone, which is basically amphetamine with a beta ketone. If you add a methyl group to amphetamine, you have methamphetamine. If you add a methyl group to cathinone, you have methcathinin, which is the street drug jet. And then once you start adding some things to the benzene ring, you can have all of your synthetic cathinones. If you add a methylene group and 2 oxygens, you have methylene diacetymethamphetamine, which is MDMA. There are dozens of different synthetic cathinones that are fairly easy to make by people who know how to make them, where they just add 1 or 2 things. All the 2C derivatives, they're basically taking a phenylethylamine structure and adding things at various carbons. So, but whether they would be able to market them legally, maybe, you know, whether there was a market for them, that's a whole different thing, but it's easy to make, and it has been done many, many times.
That is fascinating. Thank y'all. If no one has anything else to add, we will, just a friendly reminder, we will draw and post the winner of the prize tonight. So if you asked a question, your name will be put into our drawing. So keep an eye on our Facebook event page to see if you won. Does anyone else have anything to add before I continue?
I had one quick mention. I forgot to do it when I started my PowerPoint. Almost all of the photos included were credited to Alan Rockefeller, and a few were taken directly from papers, but I wanted to give credit.
Thank you so much. So yes, keep an eye on our Facebook page to see, or our Facebook event page rather, to see if you won a prize. And thank you so much to our incredible speakers for such a far out event. We learned so much about mushrooms and many other types of drugs today. We'll be posting the event video soon in case you missed part of it. And the Facebook Live event the video should still be available after we end tonight. Taste of Science does not condone any illegal activity, just to throw that out there. And check out more of our festival events running now until May 1st. On May 1st, we're going to do a trivia night and a themed cocktail that you can make with us at home. We'll be posting the recipe for that in the next couple days. And there's an event tomorrow as well called Let's Take a Selfie, the highs and lows of cell research, which will start at 3:30 Pacific time, 5:30 Central time, and 6:30 Eastern, since we're sort of all across the board here. And we've also posted a festival passport on our main Taste of Science social media. Houston will be sharing that again as well. You can win a prize for attending more events, so make sure you download that passport and keep stamping it as you attend. And you can find us at tasteofscience.org, on social media at Taste of Sci and at Taste of Sci HOU. Our Houston chapter's podcast called Tosh After Dark is at anchor.fm/Tosh After Dark on Spotify and Google Podcasts. Do any of our speakers want to share their pages or socials or have anything to plug tonight? Good. Okay. I just wanted to make sure. Like I said, check out tasteofscience.org for the rest of our festival going now through May 1st. And thank you all so much. Thank you to our phenomenal speakers. And we'll see you next time.
Thank you.
Take care. All right. Thanks, guys.
Yep. Thanks, everybody.
That was fun.
Thank y'all so much. I learned so many
Thank
things.
y'all. Have a good evening.
Your fight or flight response.
Taste of science.
The tectonic weapons.
Nuclear weapons.
Taste of science.
Earthquake gun, I guess? Why is it on the internet?